Hello viewers, do you know our DNA is constantly under attack from UV ray to chemicals, countless factors damage it every day. But nature has its own instant fix, the DNA repair mechanism, a molecular undo button that corrects damage right on the spot without cutting or replacing anything. So stay tuned till the end because once you see how this tiny repair system works, you will understand how amazing your DNA really is. So let's start the video. So what is DNA repair? So DNA repair is a mechanism where cells have an ability to remove the damage and restore the original condition. There are two types of DNA repair mechanism occurred in the cell. One is directly repair mechanism where repair occurs without breaking the DNA backbone, and another one is indirectly repair mechanism. These types of repair mechanism involves breaking and resynthesizing the DNA strand to fix lesion like double-stranded break. Mismatch repair and photoreactivation repair is an example of the directly repair mechanism, and base excision repair and nucleotide excision repair is the example of the indirect repair mechanism. This is the basic steps of the DNA repair mechanism. Both directly repair and indirectly repair mechanism follow these all 5 steps. So first step is the identify the damaged part. After identification, the repair proteins or enzyme machinery recruit in the damaged part, which removed the damaged part from the DNA strand, and next resynthesization of this damaged part occur via DNA pol 3, and next sealing occurs with the help of ligase enzyme. So both repair mechanism follow these all 5 steps. In this video, we only discuss the directly repair mechanism. So first one is photoreactivation repair. So in this repair mechanism, only thymine dimer is repaired. This thymine dimer was formed by UVB ray. The enzyme involved in this repair is the photolyase. In E. coli, this enzyme is called the photolyase enzyme. Photolyase enzyme or photoreactivation repair enzyme absorbs a photon of light and is able to cleave the bond forming the thymine dimer. So this bond is cleaved, and once the bond is cleaved, DNA is back to the normal. So this repair mechanism mainly occurred by the enzyme which was called photolyase enzyme or photoactivation repair. Next, the important directly repair mechanism that is mismatch repair mechanism, where the enzyme mutase dimer. So this is the enzyme mutase dimer detects the mismatch repair. This enzyme has an ATPase activity. Mutase complex recruit mutal which activates mutate, an enzyme that causes an incision or nick on one strand near the site of mismatch. This nicking is followed by the action of a specific helicase enzyme which was known as UVRD and one of the 3 exonuclease enzyme. The helicase unwinds the DNA and the exonuclease progressively digests. This action produces a single strand gap which is then further filled by DNA pol 3 enzyme. So the question is now, how does the bacterial mismatch repair system know which of the two mismatch nucleotides to replace? So in bacteria, mismatch repair is the based on the process of DNA methylation, which labels one strand. So DNA methylation occur in the one strand. The E. coli enzyme DAM methylase methylates only adenine residue on both strands of the sequence 5' GATC sequence. So in this GATC sequence, this adenine part was methylated by the enzyme DAM methylase. Later, the mutate protein binds at such hemimethylated sites, but its endonuclease activity is normally latent. Only when mutate is contracted by mutL or mutase located at a nearby mismatch does mutate become activated. So mutate activated when it comes to the contact with mutal and mutase. This type of repair mechanism also found in eukaryotic cell also, where the cells mutase homologs occur proteins called MSH protein or mutase homolog, and mutate homolog, also called MLH and PM, though eukaryotes have multiple mutation-like proteins with different specification. So for further know about the repair mechanism or for further classes, like and subscribe my channel. Thank you.